Hi everyone, my name is Patrick Bender, and my comps topic is about machine learning and computational chemistry, and it's specifically focused on protein structure prediction. So this is a continuation of my talk last semester, which was about machine learning and computational chemistry generally. Um, and a quick review of what I talked about last time. So I talked about sort of computational chemistry and why we care about it. The short of that is it allows us to predict things without actually making any chemicals or doing any actual wet lab analysis, which can be useful. We talked about current techniques and their limitations. And we talked about machine learning in general, and then later its specific applications to chemistry. So a quick review of what machine learning actually is. This is a basic neural network. It's composed of layers, each of which sort of acts as a little like neuron in our brain. And what those do is they take the inputs from the previous layer, in which case it's all these x's here, um, and then multiply them by some weight that's associated with each neuron. And then it's going to sum them all up, pass them through some sort of function, and then set a new value. So in this case, this neuron labeled j would be y sub j. Then that would go all the way down the line, and then continue to the next layer, and then the next layer. We can have as many layers as we want in here, and we eventually get some output layer. So what we're going to be talking about later is sort of a twist on that. What's called a convolutional neural network, which we'll see later. It's sort of a similar principle where we go sort of forward. We have this input layer, we have a bunch of layers, we have this output layer, and then if you'll remember last time, we sort of evaluate what that output layer is. So we say, okay, we know the truth from this training data. How accurate are our predictions? And then we can go back and adjust these weights a little bit. Um, and that will, if we do that, enough times, thousands or millions of times, eventually that's going to get us to a really good approximation of what we want. And you can sort of think of all machine learning models as basically just really fancy regressions. Um, so in this case, this is a two-dimensional regression where we have some initial guess, which is one of these gray lines, and then we measure the root mean squared error, which is sort of the distance between like this gray line to this particular point, and we do that for every point. And we try to minimize that. So we adjust it a little bit. Maybe that's like an overcorrection. So we adjust it back, and eventually we settle on this sort of red line here. That's sort of a good approximation of our function. And that's just sort of what machine learning does in general. So a review of what we want to do with this machine learning, we want to do sort of two things. One is to predict properties, which is if we have a particular molecule, so say we're investigating some sort of drug and we want to see if it binds to, a particular protein. We can, instead of, getting that protein and getting that drug and sort of doing a bunch of tests, instead we can simulate that drug and that protein and see what properties it has. Another thing that we can do with it is say we have the protein and but we don't have a molecule yet. We want to find what molecules could be suitable candidates for testing, and that's called inverse design. And so there's sort of four examples that I talked about. One is drug discovery, another is use in material science, catalyst discovery, and then also protein structure prediction. And I was going to choose one of these four. I wasn't really sure what at the beginning of the semester. And then all these headlines came out about Google DeepMind's AlphaFold, which is made the choice very clear for me. So we're going to be mostly talking about this, its architecture, how it works, all that sort of stuff, because I imagine this is going to be very useful in the next 5 to 10 years. Um, and so yeah. So AlphaFold specifically competed in this thing called the Critical Assessment of Techniques for Protein Structure Prediction, or CASP, which is a open competition for any protein researchers, or anyone can do it. It's basically a competition to see who can get the best algorithms and who can do the best protein prediction. So AlphaFold got 25 out of 43 proteins correct. And the last one, the next closest from the competition, got 14. The global distance score, which is basically a measure of how accurate is this, they got 63. And it needs to be around 85 to 90 for real world applications. Now, I will note that this was compared to people who actually partook in the competition. CASP 13 happened in 2018. That's who they're competing against, basically. Um, So it was not a live results, but CASP 14 is actually happening right now. Registration has already happened by now, and the target proteins should have been released by the time you see this. They said sometime in mid-April. I think they're actually releasing it on the 15th. And hopefully, if coronavirus isn't a thing anymore, the results will be shown at a conference in Europe in December. So this is the results that AlphaFold generated. So a quick explanation of what these are. These are called distograms. They're basically a measure of the distance between two amino acids. So like, say this is the first amino acid, and this is like the last. So we see that the obviously amino acids are very close to themselves, which is why you see this sort of stripe. But we can compare the first amino acid to the last amino acid and say, okay, that's not very close because you get the brighter, more intense green colors the closer you are. These are actually, sorry, the first and the last amino acid for this one is very close. The one for this one is not very close since it's very dark. And you can see that this column is the ground truth. So this is what has been experimentally determined by X-ray crystallography and other techniques. And these are the predicted results from AlphaFold. And as you can see, they're very close. And here's a visual representation of what they are down here. So this is what they would look like in real life. And as you can see, the green is the ground truth, and then the blue is the predicted structure. And as you can see, they're very close, which is very impressive. So yeah. So a quick overview of what I'm going to be talking about in this talk is I'm going to be talking about current techniques for protein folding, um, and then I'm going to be talking about spending a lot of the time talking about AlphaFold's architecture, which sort of comes in. There's sort of three parts to it. The first is the feature extraction, which is basically taking the raw data and filtering it a little bit to get more useful information. Second is the convolutional neural net, which I talked about, which is sort of the heart of it. And then there's the gradient descent, which is sort of a further refining stage that allows us to have a more accurate understanding of what exactly the protein will look like. So first, the current techniques. A lot of them rely on this thing called fragment assembly, which is basically we have some knowledge about proteins, and then we look for similar structures in our target protein. So, say we're looking at like some sort of oxygen binding blood, protein, like sort of similar to hemoglobin, but some maybe find some novel creature or something. We can look and see, okay, we have lots of information about hemoglobin, about myoglobin, stuff like that. We can probably find fragments that are similar to that protein in, known proteins. And we can sort of match those up and see what exactly, how we can have some guesses about what that protein will look like, what parts of it sort of have either, pleated sheets or helical structures, how it folds, stuff like that. And there's sort of two methods. First is called Cho-Fassman. This was in the 70s. It's entirely empirical. And the second is called the Gore method, which is sort of an extension on Cho-Fassman, but includes a lot more information theory and sort of Bayesian analysis. Um, so to get into those, we have Cho-Fassman, which is primarily focused on the secondary structure prediction, which, for those of you who've taken biology, is proteins sort of have amino acid strings, and they sort of curl up into either helixes or sheets, or they have turns, and those are the secondary structures. So it primarily focuses on those. It uses empirical parameters. So they had back in the 70s, they had a bunch of these proteins that they knew about. They knew which amino acids were in helixes and which ones were in sheets or which ones were in turns, and they sort of assigned a probability to each one. It was only about 50% accurate. It wasn't really based on a representative sample of proteins since they didn't really have a whole lot at the time. And it overall wasn't really effective. Basically, it's mostly just guessing. If you have like a lot of amino acids that are likely to form helices, you can have a reasonable guess that it's probably a helix there. But but it's better than nothing basically. And then the Gore method makes it a lot better. It is similarly empirical in that it does that sort of statistical analysis, But instead of just looking at each individual amino acid, it takes into account the neighbors of each amino acid. Actually, it goes 17. It accounts for 17 amino acids in total. And it used this 17 by 20 scoring matrix that's sort of slid along the amino acid string, and would tell you sort of give you a probability that a particular amino acid is in a particular secondary structure. And from there, you can have a sort of look at it and reasonably guess which parts were helices and which parts were sheets, etc. So now we're into AlphaFold, which is not the first neural network-based protein structure predictor, but it is definitely a very large step forward in sort of its complexity and sort of accuracy. I mentioned before there were sort of three main parts. The first is the MSA, multiple sequence analysis feature extraction, which I'll be talking about later. But basically, we had the sort of raw data of just amino acid sequences, which is not very useful, and we want to sort of get some more concrete and workable data. Then we have the deep neural network, which is going to give us the histogram that we saw later. And important to note, I'll be talking mostly about distances, but a lot of this stuff also applies to torsion angles. So that's the angles that amino acids form with each other. So this is an example of the histogram that you saw earlier. And the last part is the gradient descent, which you can see here. Is sort of we have this initial guess, and we sort of do some simulations to get it to fold properly. And you can see that it actually this step is actually very important, even though it's very simple because it takes the basically the accuracy score from zero point 0.2 all the way up to 0.7. Yeah, so that's the basics. I'll be going through each sequentially, and yeah, so let's get into that. So first thing is feature extraction. So the basic premise of this is that raw data is really hard to work with, so it needs to be sometimes filtered or transformed. So in the case of AlphaFold, we start with basically just a bunch of amino acid sequences. And while machine learning is pretty powerful, it still needs a little bit of help, especially getting started. And it's hard to go from like completely unrelated data sets that if the inputs and outputs are completely unrelated, it's kind of hard to go from one to the other. So instead, we want a way to turn that amino acid sequence into like distance-based data, since our output is sort of distance-based. Another way to think about this is dimensional compression or clustering. Basically, it's a way of extracting important information without because in a lot of data, things are redundant, or things are correlated. So you just want to get the parts that really matter. And what exactly that looks like, because that's a very broad description, and what that exactly looks like varies by experiment. There are, lots and lots of different types of feature extraction, but I'm going to be talking about one specifically as an example, which is principal component analysis, which some of you have probably heard of before. And then for AlphaFold, I'm going to be talking about multiple sequence analysis, which is what they use. So PCA, basically, you can see what I was talking about earlier with dimensionality reduction, because you have this sort of three-dimensional data set here, and then you want to map that onto a two-dimensional space. So we have, all these sort of clusters of these, little red crosses, those little blue X's, and it's kind of hard to visualize what exactly is going on here. You can sort of tell, it's like, okay, there's sort of these clusters here, but it's sort of hard to see exactly what's going on. And it's impossible to see if you have, more than three dimensions, which this data set definitely does, or the AlphaFold data set definitely does. So you want to turn that into the sort of two-dimensional space. And basically the way you're going to do that is you're going to find out which way is the principal component. And that means, so principal component one is the direction that you're going to change. If you go in that direction, it's going to change the most of your output. Basically going this way on the component space is going to change a lot what your result is. So it's going to change, the change in input is going to rapidly change the output. Principal component two is sort of the secondarily, sort of what's second most going to change. This will come up later in gradient descent. It's basically what parts of the data are important, basically. And that's a very hand wavy description of it. But there's a lot more of the deeper mathematics that I that we really just don't have time to get into behind this. So multiple sequence analysis, which is what AlphaFold actually uses. So our goal is to find out which amino acids are in contact, or how far apart things are, because our output's in distance, so we want our input to also be sort of a similar metric. Um, so basically what MSA does, it compares genetically related organisms' amino acid sequences. And the reason why I want to do that is because if amino acids that mutate together, they're likely to be close to each other. And the reason for that is, so for example, if we have some like polar amino acid, and that's in contact with some other polar amino acid. But if one of them, mutates to be a nonpolar amino acid, that's likely to disrupt the protein and substantially sort of compromise its ability to function. So that's less likely to be passed on. So, but if they both mutate to be nonpolar, though that is more likely to be passed on. So we can tell which parts are in contact, which parts are not within a reasonable accuracy. The more sequences you look at, the more likely it is to be true. And MSA is basically just an algorithmic way to determine that, because we can look at, just a bunch of sequences and sort of line them up, but that's not very scalable and a little hard to do. And this is just, cleaner. So here's an example of what multiple sequence alignment looks like. So we have like human DNA and mouse DNA. I think these are ribosomal proteins. So we can see that these two are very similar because, obviously us and mice are relatively genetically similar. But we can see here, I don't know exactly what this organism is, but we can see that this P changes to an S. But nothing else here really changes. So we can be reasonably sure that this S is either, we can compare it to the P, and sort of compare the actual amino acids. And so, for example, if like S was like polar and P was also polar, it might be in contact with some other thing. But if this was like completely different, we could be reasonably certain that none of these other things were really in contact with this. So this would sort of be on its own. And we can do that for all these different ones. And the more sequences, and as you can see, like all of these sequences, you can get a pretty good picture of what's going on if the more sequences you have. So now we're on to convolutional neural networks. So it just takes that data that we get from the feature extraction and turns it into a rough guess of what the distances are. So a reminder from machine learning, we have this sort of forward pass, which is going all the way through the neural network. Then we're going to evaluate how well that network did. Then we're going to do backpropagation and reassign the weights, which is basically this sort of step is the neural network learning. So the major difference is how the forward pass works and how sort of the neural network goes forward. It is inspired by the visual cortex. So the thing about the visual cortex is that in certain regions of our visual field, some neurons only respond to light in that field. So this was originally used for image processing, but it turns out it has a lot of different uses, and you'll see why exactly this sort of visual cortex and the region and the visual field region is a useful analogy for thinking about this. So this is sort of the basic operation of a convolutional neural network, this image on the right, or on the left. This little matrix is called a kernel, and this is going to be our input data. So this is like our first layer, for example, and then this is going to be our second layer. So it's going to compare all of these elements of the matrix. It's going to multiply them together, and then it's going to give us this sort of little output matrix. I think this one right here should be 160. I think the person who made the image just messed up on that. But then we're going to sum them all together to get a single scalar number, and this is going to be our output of that first layer. So you can sort of see an analogy between the weights. So whereas before in our previous neural network, the weights were scalars, in this case the weights are going to be this square matrix. And so it's going to sweep through the data layers. So in this case, it would go from here to sort of here next, and then go one over, and then one over, and then it would start back over here. And you may notice that this is going to actually give us, if we just did this, it would actually give us a 3 by 3 output. So one thing that people do sometimes is pad pad the input matrix. They only did it on this side, but they would just pad it with zeros, just to keep it the same size. Um, so yeah, so that's sort of the basics of what a convolution is. And if you stack all these layers together, you sort of get a similar effect to the traditional neural network that we talked about earlier. Um, so in this case, the input is this L by L square matrix, which L is the sort of distance or the amount of number of amino acids that a protein has. Um, and it uses the 64 by 64 kernel, which was just sort of experimentally determined. I think they tried a 128 by 128, and they just really couldn't get it to work. And then it goes through 220 layers, which is a pretty substantial neural network. And then it's going to give us both distance and angle predictions. I'm mostly kind of talking in the context of distance because that's sort of the easiest to understand. But yeah, and then it was trained for 5 days on 8 GPUs. So now we have this sort of rough idea of, or this rough guess of what this protein looks like, but we still need to further refine it. So to do that, we use a thing called gradient descent. And you can the easiest way for y'all to think about this is just it simulates potential energy wells. So it's going to bring the protein into a more stable conformation, a sort of lower in the energy well. And this applies to both torsion angles and distance because the torsion angles are actually pretty important for the potential energy. And it uses this thing called a noisy restart to stop it from getting stuck in a local minimum. And hopefully you'll get a better idea of what that looks like. And also in the architecture picture, which I'll show again, it has examples of what gradient descent actually looks like. So in this case, this is gradient descent in 2 dimensions. So essentially what we do, so you can think of this A right here, this is our starting point. And we're going to look at basically which direction to go in to get the area of, or the direction of greatest descent. In 2 dimensions, this is super easy. In 3 dimensions, it's also actually mathematically very easy. It's a little harder to visualize, but sort of you can very easily analogize this to multiple dimensions. But basically, we look for the direction of greatest decrease. In this case, it's going to be the left, and then we take the derivative at that point. In multiple dimensions, it's called a gradient, but it's essentially the same thing, which is why it's called gradient descent. And then we step forward some size. So in this case, it's like, let's call this like 1. And then, so we step forward 1 times the derivative, and then we're at this new place. And then we evaluate the derivative again at this particular point. And then we just keep going, until eventually, at some point, we're going to get to a local minimum. You can also, to note, you can also do this to find a maximum. You just reverse the direction of the gradient. So I also mentioned before the noisy restarts. So imagine instead of just being a smooth well here, it actually dips up back a little bit and then down. And then this sort of region down here is the actual, the lowest potential energy. What a noisy restart would do is say, okay, we've reached this local minimum here. Let's like jump just a little bit this way. And then go from there. And that's going to give us our potential energy, which you see a lot in these sorts of gradient descent algorithms. These noisy restarts are very important for finding a global minimum instead of just finding a sort of a local minimum. Yeah, so that's basically gradient descent. You can sort of, I hope this makes sense in multiple dimensions. It's kind of hard to show it. But this is basically what's being applied to thousands of dimensions. And the function in this case, the sort of gradient, is all the forces that a particular amino acid experiences. So say it's very close to something, another amino acid that was attracting it a lot, that would pull it in that direction as opposed to away from it. And you sort of just simulate, it's basically just a simulation of what the physics looks like, which hopefully you can see here is makes a little more sense because we have this sort of protein that's all strung out, and then maybe this part is attracted to this part. So this swings over a little bit here, and then this sort of pulls in. It's basically just sort of simulating what the forces that the protein experiences. So in review of this whole thing, we have all of the amino acid data or the amino acid sequence. Then we're going to extract some features using MSA. That's going to give us a bunch of a bunch of distances. Then we're going to use the convolutional neural network. And then we're going to get out this histogram, which is the distance and torsion distributions. And then we're going to use this gradient descent to make it more accurate. And yeah, so that's basically all of AlphaFold. A quick note before we go: this is actually not just a bunch of academic nonsense. It was actually useful because experimental protein modeling takes a while and could also, frankly, be dangerous in the case of coronavirus. Um, and Google DeepMind applied the AlphaFold technology to coronavirus and found that their prediction to the spike protein, which you may have heard about, was accurate to the sort of experimentally determined one. So they took all the amino acid sequences that they could find and for ones that had no known template, which I talked about earlier. So these are sort of novel proteins that people have never seen before and have no really frame of reference, and sort of did a bunch of things, a bunch of simulations of that, which is called free modeling, where you don't have those templates before. So yeah, so they were able to get some predictions of what the coronavirus, some of coronavirus's proteins, which hopefully could be useful in the fight against the global pandemic. Um, yeah, so here's a link to the paper. Yeah, I'll also be linking one down below if this is a YouTube video. Um, yeah, so it's a very interesting paper. It's pretty short. I would advise you take a look at it, especially if you are more into the math and computer science parts of it. But yeah, that is all. I hope to be seeing your questions on or listening to your questions and answering them on Friday. Yeah, that's it. Take care.